Okay, so today's session is nematode and viral pathogens. So I've lumped them together because we don't see a whole lot of nematode disease. We don't see a whole lot of viral disease in the whole grand scheme of things, surely not compared to some of the other groups, the fungi, oomycetes, and bacteria. So I put them together and I've got some samples. We can't cover all the different viruses and all the different nematodes. So we'll just look at a few just for fun. We'll start with viruses. Again, if anybody has any questions, come into the chat box and I will try to answer. I'll try to catch those as soon as possible, but it's okay. I can always backtrack. So let's start with viruses. What are viruses? Some unique things about viruses that are really important to remember is that viruses go systemic. When a plant is infected with virus, it is infected, the entire plant is infected, and it's infected for life. Viruses cannot be managed or suppressed or controlled in any way by pesticides. So basically, you're in trouble if a virus comes in. Viruses are spread by several different means. There are insect vectors and there's mechanical transmission. I'll talk about that in a second. Also with viruses, there's a lot of variation. Okay, backtrack. Viruses replicate in the plant and they must be inside the plant in order to replicate. So they basically hijack the plant into replicating their DNA or RNA for them. So they're very, very simple, yet they're very clever. And so it's the actual plant nucleus that is replicating viruses. Once they're there, again, they're there for life. So virus management is usually just trying to stop spread, roguing plants and stopping spread. So with viruses, there are a couple of common ones, and then there are some that we just rarely see. And so I've just got a couple of really common ones for us just to talk generally about viruses. So let's see where we're going to start. Oh, I know what I want to do first. So I have TMV right here. TMV, let's see, let me get my stuff. TMV is tobacco mosaic virus. It is a tabamovirus, and TMV affects about 100 or 125 different species in the plant world. TMV is mechanically transmitted, and I want to talk about mechanically transmitted viruses. First of all, I should be wearing gloves. Shame on me, I did not go down to the lab to get gloves. I'm going to come down here. I should be wearing gloves. I have a TMV sample here on paper towel, and there is a fruit infection here. I don't know, probably one of you recognizes this sample. I got it from the diagnostic lab. So a mechanically transmitted virus is one that can be transmitted by any touch. So any movement of plant sap or plant material. A TMV can, or any mechanically transmitted virus can move on our hands. TMV is very persistent virus. So it is now on my hands. I've just touched my pruners. If I take those pruners home or take them outside to do any demos, I'm spreading this virus. So before I leave today, I really need to do a lot of cleanup. So if you're suspecting viruses, and I remember a couple of years ago, new agents that came through orientation got kind of a field kit that included like the paper shoe covers and gloves. This is the time that you should wear gloves if you suspect TMV in any way, shape, or form. When I'm finished with this sample, it's going to go back in a bag, but I really need to do a lot of cleaning. I was handling it yesterday, and here I am handling it again. So mechanically transmitted viruses are moved by the touch meaning us, touch meaning all of the tools that we take in and out of the field. It can move by any of your tractors or equipment. Even leaves touching each other when they're blowing. It can move from plant to plant by any kind of touch. So when we say mechanically transmitted, not just mechanical, but any touch transmission. TMV in particular, again, is quite the widespread virus, very common in tobacco and any solanaceous crops. That's where we see it the most. The symptoms are pretty much a mosaic That's why we call it tobacco mosaic. And with viruses, really knowing some of the symptoms, I have not updated the vocabulary list. I will do that when we log off today and make sure it's on the training website. So mosaic, think about the definition of mosaic. Mosaic means a change in like an angular or a non-pattern change in color. And with some of these viruses, it's really not as distinct. So you can Google image it, and you're gonna see very classic, beautiful symptoms. It's not really that easy in the field. So I'm gonna use my Dino Light and see if I can show you some of these patterns of the kind of the yellow, light green, dark green in this mosaic pattern. I'm also gonna show you a fruit. Let me see how, this is a tomato fruit. And you'll see that it is not the classic symptom that I'm describing. So just at this point, it's probably just, hey, something's going wrong. So we've got, and I've got a smaller one that might have a little bit better of a pattern. So I'll do that with my Dino Light, and we will see if we can get something a little bit clearer for you to see what a mosaic pattern really looks like. So this is going to be a broad view. We'll see what we can get here. The joy of doing this live, right? So I'm going to share my screen. There we go. I'm going to share my Dino Light screen. My Dino Light on? Yeah. Bear with me here. Oh, that came out pretty good. So this is a mosaic. I'm going the wrong way. These are some mosaic patterns. And you'll see that you've got no pattern at all. All right, so the symptoms are pretty, I'm sorry, I know I'm jerking around. The symptoms are pretty much, so you'll see very dark green, probably excessively green. You'll see normal green, and you'll kind of see yellow. There are not really any spots on these leaves. Let's see if I can focus just a little bit better. There are not really spots per se on these leaves, so it is just a variation in color. You can see that. And with TMV, you also see some distinct yellowing sometimes of veins. And you can see that, let me get my pointer. You can, oh, no, sorry. You can see that starting to happen here. The yellowing of some of the veins. Sometimes that'll start a little bit earlier. And then you'll kind of see, like the roughness going on in this leaf. This leaf isn't nice and flat and normal. Remember in the first couple of sessions, we talked about knowing what a healthy plant looks like. And this is kind of one of those that you really need to know what a healthy plant looks like. There we see again, you see kind of that, that modeling or that mosaic. I'm sorry, not modeling. Modeling is a different symptom. You'll see that, you'll see kind of some puckering almost and some leaf distortion going on. So this isn't quite right. And that is pretty common in TMV. And I'll just kind of cruise around here. Bad driver. It's kind of like backing up a trailer. Everywhere you want to go, you got to do it the opposite of what your brain tells you. Okay, let's see. I'm trying to get it centered, and I'll stop for you. So this is a very clear mosaic. So just kind of the variation. Any questions there? By the way, when I'm sharing my screen, I can't really see your comments, so there's always a little bit of a delay when I'm checking, or Kim helping me check those. So if you have any questions, or if you want to see this a little bit better, let me know. If you just kind of Google TMV, make sure you put virus, because there are even some New York Stock Exchange ticker symbols that look a lot like the virus abbreviation. So that can get you some really interesting things. Questions? TMV, mechanically transmitted viruses are the most dangerous of those for us as we walk in and out of fields and really risk the movement. Mechanically transmitted viruses can also move by touching leaves, moving plant material, propagation material, getting clean plants coming in. I saw a blinking light. Okay. How long does tobacco mosaic survive? Oh, you know, I'm going to have to look that one up. I don't work with any crops that are common with TMV. I will find that out for you, April, and I will get back to you by the end of the session. So sometimes they can be persistent for days. I think TMV is weeks or months. TMV is kind of spreads like wildfire. This is one, like, I would love to have this one for you for the practicum. In fact, it was kind of my plan. I was going to give you TMV at a really low level for you to diagnose, but, you know, I just can't move it. It would be unfair for me to show up in your county with TMV and you have it all over and somebody walk out the door. So, but I will check on that for you and I will verify. Okay. You see a little bit of kind of distortion at the base. One thing about viruses, one thing that we look for when we're diagnosing viruses is that we're always aiming for the youngest leaves. So virus symptoms usually start in the youngest leaves. Usually when we have fungi or even herbicide, kind of everything gets it equally. Whereas viruses, they'll kind of initiate in that new growth. And that's always kind of an indication that, hey, something is wrong. Does anybody want to see any more of this? Let's see where I'm going. Okay. All right. That's TMV. One thing I do want to show you, I'm going to flip back. I'm going to stop sharing my screen here. And I want to kind of go over something. So do you know when you send a sample to the plant disease diagnostic lab, your report will come back that that they diagnosed by idea strip or by ELISA. So those are some terms that you may see in your diagnostic report and you don't know what the heck they're talking about. So I want to show you one of the idea strips. We'll do it with TMV. I have a TMV kit right now, and I'll show you exactly kind of what goes on. An idea strip, what did I do with the strip? Here we go. The idea strip, idea is a company, so idea is nothing magical. And it is just basically a dipstick. It is loaded with antibodies that are specific to, in this case, this one is TMV. It's kind of like a pregnancy test. It is or it isn't. So what's going to happen is I am going to macerate some tissue. The little green section there is going to be what goes into the sample, and then it will wick its way up. 5 to 30 minutes is what it takes for these strips, and they just light up. There's a line or there's not a line. So I'm going to do that real quick, and I'm going to show you how I do it because it's like really that easy. And then we're going to just put it aside and just wait because we should have a positive. One qualifier is that this is an expired strip. They're usually viable for about a year, and these are some expired strips that I've got because I did not want to use any of the strips from the diagnostic lab. So I kind of swiped this one. So it may not work. So what we do with the strip, with the kit, is we start with this little bag. This little bag has some buffer in it. I don't know if you can see kind of some liquid rolling around there. And there's a mesh screen inside. The mesh screen is basically to macerate the tissue. So I'm pushing all of the liquid down to the bottom because you don't want to lose any of the liquid. And I'm basically taking scissors and I'm cutting the top off. And there are 2 pieces of screen in there. So I'm separating that out with my hand, and I'm going to put a piece of tissue inside. So I will, so I've still got my sample here. I'm going to cut a piece about 1 inch by 1 inch. Too much sample in here will overwhelm the system. Okay, so 1 inch by 1 inch is a sufficient amount of tissue. So here I am, and I'm going in between these pieces of mesh, just dropping it right on down. See, it's in, let's see, sorry guys, see, there's the buffer. And I'm going to just kind of hold this. All right, you get my table view again. And I will take anything and I will macerate the tissue. So this is just my ink pen. Macerating this tissue. So what we're doing is we're putting all of the plant tissue and, in theory, the virus in suspension. And so here we are. We've got green water, green buffer. All right. So I'm opening the bag back up. I'm letting all of the liquid go down. And I will place my strip. I will place my strip green side down right into the sample. There we are. There it is. And I will wait. So I'm going to just set it right here while we move on. And in a few minutes, oh wow, it's moving quickly. In a few minutes, we will see what the results are. What I forgot to show you is the fruit sample. So I'm going to share my screen again. Okay, I'm going to share my screen again. Back to this TMV. Here is a fruit, a tomato fruit that's infected. And we don't see this real often. So we'll see kind of some. Let's see if I can use my blade. I'm holding this with my hand, so it'll be a little bit unsteady. So we'll see kind of some abnormalities. We'll see chlorosis, necrosis. Sorry, I know it's blurry here. I guess this is not the best idea. So you'll see just this discoloration. So you really are seeing mosaic on the fruit. And then here is the larger fruit. If you can back out a little bit, it's a little bit harder to see mosaic. So the fruit is probably what alerted this grower into something being wrong, but I would not look at this sample and determine that it was TMV. You see that? I mean, it's an odd one. Viruses are hard. Let's see, we can look a little bit more here. Sorry, I know you're probably dizzy by my jerking hand. So this is not very classic. This is not what I would say, hey, those are typical virus symptoms. All right, so there we go. TMV, tobacco mosaic virus, and it is mechanically transmitted. Oh, by the way, here we go. I'm going to do this. And you can see. No. No. There we go. See the 2 lines? No, this isn't a very good idea. So see the 2 lines? The sample is showing up upside down. I'm going to switch this off. I'm going to get you back where I should be. Sorry. All right. So here's a sample very quickly. You'll see there are 2 lines there. The bottom line is, which one is the bottom line? One is the control and one is the positive. Do you remember, Kim? I think the bottom one is the positive. There are 2 lines there. The bottom one is the positive, the upper one is the control. Okay, so basically the control tells us that it worked. The bottom line is the positive. There are 2 there. I can photograph this if anybody really wants to see it. So there's a positive line here, there is the control. So what you need to know when you're doing this, if the control doesn't light up, it means the kit did not work. And again, this was an expired strip, so I wanted to make sure. And then we have a positive. Now we had very classic symptoms on that leaf, so we got a really quick positive. If indeed we had a very low titer, and titer means concentration in virus talk, then we would have left it there that entire 30 minutes. So it can take 5 to 30 minutes for the kit to actually detect virus. The lower the titer or the lower the concentration, the longer it would take. So we got a positive right away because this one was classic, this one was easy. They're not always that easy. And if you realize that in the diagnostic world, sometimes you'll get a report and the diagnosticians will indicate all the different things they checked. So they go through an assortment of these just to sometimes eliminate what might be an emergency or a severe situation. And sometimes they do it because the symptoms really resemble certain viruses. So any questions there? I'm happy to revisit it. I'm going to set it aside instead of tossing it in the garbage. All right, so we're going to move on to a different virus. And this is one that it's kind of new to me because I don't do tobacco. This is tomato spotted wilt virus. It is here. It is on tobacco. Tomato spotted wilt virus is a very broad host range. Over 1,000 species of host plants are affected by tomato spotted wilt. And here we have it on some tobacco samples. I guess I need to move my angle here again. So I've got 2 small plants in the tray. Not looking really good, but you have some normal yellowing on tobacco anyway. So 2 different plants I want to show you. TSWV is, so notice all of our viruses that we always abbreviate that way. So this is tomato spotted wilt in tobacco. You'll see we've got some necrosis along the veins, which is a very classic symptom of the virus. And then you've got kind of this puckering or this roughness here, and I will get this closer for you. You see, so it's not flat, it's kind of rough there, right at that base. Very classic. And look down the veins. You'll see that necrosis kind of following up those veins. That is classic of this particular virus. Let's see, let me move some things around here for you. The puckering, the twisting of the buds isn't, oh wait, I had a ring spot. Okay, we have an occasional ring spot. Can you see it? There's a ring spot on that sample right here. I know, these camera angles. So there's kind of a ring spot, and that would be another distinguishing symptom. But this roughness here, then we'll also have roughness in buds. So I'll show you several different samples, several different parts of the sample. I'm going to dismantle this plant. There we go. We've got the roughening here. You see it's kind of scrunchy and puckered. Then we've got necrosis along the vein. And you can kind of see that puckering there. So tomato spotted wilt virus is transmitted by thrips. And so most of your viruses are vectored by an insect of some sort. And this one is transmitted by thrips. It's pretty ubiquitous. It's everywhere, and thrips are going to carry it around. The virus causes stunting, as you can see with that puckering. It will cause some ring spots on occasion. So I'm dismantling this small, this bud. And again, we've got those classic symptoms going on, the puckering at the end. And I will try to get you into this bud. We can see some, hopefully we can see some twisting. So some rough, I wish I knew how to focus this thing a little bit better for you. We'll see some twisting within that bud. Maybe I need to use my Dino light. Oh, I'm taking pictures again, darn. Okay, did that pop up? Can you see a little bit better? Let me try something else for you guys. So there's some necrosis there. There's some twisting. There's some puckering, so you can kind of see that. So you're taking a whole range of symptoms and you are kind of identifying. Now, if you are an agent who has a lot of tobacco in your county, you would be very familiar or make yourself familiar with some of these very common viruses. But if you don't work with them, it's not something that you would be familiar with, and that's completely okay. So if you see something that's just not normal, if you see some puckering, or you can just do a search and then you can say, hey, this is the symptom. So I'd really like you to know kind of what some of these symptoms mean with some of these ring spot, or what, like a necrotic ring spot. They don't show up real well, but there's kind of some ring spot going on there. So just kind of knowing the definition of some of those symptoms, you can kind of put it all together. Any questions on tomato spotted wilt virus? And this is an easy one for the diagnostic lab, because they use the test strips again. So they just the dipstick test or the Agdia test strip on those. So you'll see, I'm looking now, the more I look, the more I'm seeing some of these ring spots going on here, in addition to necrosis, that's a root, in addition to necrosis along the veins. So these are classic symptoms if you know what you're looking for. Questions on TSWV? And I know viruses are really hard. I'm not a virologist, and so I feel that viruses are one of the hardest things that I work on, because I don't work on them very much. All right, and the final virus that I want to kind of cover today, this is an ornamental one. This is another very common virus. This is rose rosette virus. I'm sure everybody saw when I pulled the sample out. And knowing about, and again, referring to literature, if you're not familiar with this one, rose rosette virus is a mite transmitted virus, a special mite, a special aerified mite that transmits this virus. The mite populations when there is virus are about 50 times higher than mite populations when there is no virus there, but you pretty much will always find these mites. Rose rosette virus in particular causes a few symptoms. This one's been in a bag, so you'll see it's kind of turned down on the top, but very, very exaggerated red, new growth. Now we know that a lot of these roses, the new growth is red, but this is really excessive. You see that from a distance. So it's really excessive and it's really extensive. We have a symptom here called witch's broom. It's a little bit not quite as exaggerated as it is sometimes. And we have a shortening of the internodes. Let's see. Trying to see what you see. Shortening of the internodes, so less space in between the nodes or in between the branchlets, and so you get kind of this cluster of new growth right at the top. So that's what this one is, and it can be much more exaggerated than this. One common rose rosette virus symptom is excessive thorniness. I'm going to run this by you. Look at the thorns going on in this guy. Let me see if I can get you close enough. So excessive thorniness is a very common symptom. What if I do it this way? I have a white piece of paper, so you can see the excessive thorns that are going on here. That's another symptom. We can have distortion of the flowers and buds, which is very common. And like any other virus, there are a lot of herbicide damage symptoms that look just like this. So being very careful in what you're diagnosing. So herbicide damage, especially glyphosate damage, will create all of this reddening. It will create witch's broom, and it will also induce excessive thorniness. This is a rose cultivar called Knockout. Knockout roses, remember when they went on the market, they were touted as being disease resistant, which really meant that they were resistant to black spot. Since then, they've kind of overcome that resistance, but there was nothing that said that they were resistant to other diseases, in particular rose rosette virus. It seems that if you look around, knockout roses are much more, they seem to be much more susceptible to rose rosette virus. I think what's happening though is these viruses, first of all, knockout roses are just so common that we just see more. If we're going to see rose rosette, we'll find it on the most common roses. Also propagation, viruses are moved. Remember, viruses are systemic. So if they are moved through any kind of propagation means, and a highly propagated rose, especially from cuttings, this is the way it's done. So rose rosette virus, if anybody wants to see any of that up close, just run it through the chat and I'm happy to do that for you. Any other virus questions? So viruses are tricky. The symptoms sometimes are not really clear. If you have a ring spot virus, that's always a very clear symptom. But otherwise, viruses aren't really that clear unless you really have experience. People who work with viruses can identify them lickety-split. But for those who are not virologists, it is hard. So taking lots of information down and knowing what's common on the crop sometimes will help you get much closer, at least to know when you should alert someone that this is a problem beyond a simple field diagnosis. Viruses have to be identified either by an immunostrip of some sort or some kind of molecular testing. So they would have to come to the diagnostic lab. Okay, I have a question. Coneflower, mention anything about a virus? Coneflowers would probably have a phytoplasma. Do you have a picture, Kathy, of the coneflower? Yeah, petals distorted and the flowers are green. So the petals kind of turn into kind of leaves. So that's probably a phytoplasma, which we classify with bacteria. That one can be very common. It moves through seed stalk. So definitely let's talk about that. I know a couple of agents have had problems. Just rogue it out, make sure that phytoplasma or bacteria-like bacteria-like pathogen is not spreading. And sometimes it's around, I think it's spread by aphids, if I remember right. And so it's hard to control vectors because we can't control all insects. So that's another question about viruses in general. And in this case, your phytoplasma, Kathy, is that we can't control all insects. So we can manage insects to a degree, but there's no way that we can ever completely control an insect population. So if you've got a virus, and by the way, a lot of weed hosts and a lot of surrounding plants will be reservoirs for viruses, for phytoplasma in this case. So the movement of insects is going to definitely transmit some of these pathogens. So when we always talk about sanitation and keeping your site weed free, that's one very important consideration. Now, how far do insect vectors move? Really kind of depends. Insects won't move far if they don't have to, but they can, and we know that they can move very wide. So so there's not a magic number. So if you keep your field itself very clean, some of your vectors may just choose not to visit your field or visit just the edges. Whereas if you have a very dirty field, then they're everywhere and they're just moving virus, or in this case, phytoplasma around. So think about sanitation, think about basic insect management or vector management, but not to think that there is a way to completely get rid of it. And if you have infested vector insects, for instance, if a thrip picks up virus from one plant and moves it to another, just a few vectoring insects that are infested with virus are harder to control or harder to eliminate. We can't eliminate. So a basic insect or a vector management program, weed management program, and index stock. So don't bring in anything that's potentially infected from the start. So cheap seeds, saving your seeds sometimes, moving plant material around, not cleaning your greenhouses out between seasons. You can kind of pick up that virus and you can move it from season to season or from crop to crop. Okay, any other questions?
Can you double back on the one about tobacco mosaic virus?
Oh, okay. Huh?
And how long it can survive?
Did you find? Okay, 1 second. Did you find in particular?
So they don't necessarily give a timeline, but it does say that it can overwinter. And it also talks about that it can be on seed coats, it can be made by workers, and that it can oftentimes survive in infected plant debris, perennial weeds, and perhaps in the soil. But it doesn't give a timeline.
Okay. So Kim was looking for a timeline for me for the TMV virus, I'm sorry, and how long it was persistent. As she found a lot of information about it overwintering in soil, like being in like plant debris, those kinds of things. But we haven't locked down how long it can stay viable on a hard surface or on tools or something like that. So we'll get that. So this is a viable source that she's looking at, and there's nothing hard lined. Okay. All right. I've got to move it up. I spent a lot more time on viruses than I planned to. Let's switch over to nematodes. So what are nematodes? Nematodes are microscopic roundworms. So we went from a pathogen that's not even a single cell. It's a very primitive to a roundworm that is much more complex in its life cycle. And some universities classify nematodes with plant pathology. Some put them in the entomology category. We're going to cover them today. We're going to cover them really quickly and some things that we'll probably see a little bit more. One thing we do have, this is a root knot nematode. Let me get my tray back out. I'm about to have soil. One of you might recognize this one. This one comes from Princeton. If anybody had a tomato root knot sample from Princeton, this is it. We've kind of snatched it. Brenda was kind enough to send it to us. Root knot nematodes will latch onto roots, and they will feed on roots, and the female will insist, and that's what the knot is. So when she latches on, her body will begin to insist, and she will become basically the cyst, and the eggs will develop in there. So a big knot means a lot of eggs. Root knot nematodes, or any nematodes, would be soil-borne, so they move in soil. Once you have an infestation, populations can build up if you have susceptible hosts repeatedly without any kind of rotation, for instance. That's how we manage nematodes here. We don't fumigate soils here in Kentucky. So rotations, solarization if you have a very small area, but that's how we manage nematodes. So rotating, knowing your nematode population and rotating into something that's not susceptible or less susceptible is the way we bring populations back down. But if we don't rotate, and high tunnels are really guilty for this because you can't really rotate out of a high tunnel unless you're not using high tunnel anymore. So now we have some high tunnels that are on tracks, and you can semi-rotate your land, but overall we really need a good rotation. This one in particular is root knot. I have not washed this one off because I want to show you what you would see if you were actually in the field. So I'm going to swing my dyno light around. I'm going to attempt this. So if you can just bear with me, I'm gonna. Well, if you want to see what I'm doing, sorry. So I have this dirty sample in a tray, and I'm going to just try to put it under my dyno light, and then I'm going to lower my dyno light there. So that's what I'm doing. I'm going to switch you over to share screen. Oh, there we go. So let me try to go a little bit broader. So these are just bumpy roots, basically what we're seeing. So we're seeing bumpy roots here. And of course there's soil on these roots. If you wash it off though, they're not coming off. So I'm not very good at this, am I? Oh, that's as far as we go. See, our magnification is better than I wanted it to be today. Well, I'm completely unsuccessful with you guys. So we see the knots, a lot of knots here. I think this would be so hard, right? So this is a very high population of nematodes in this soil. I don't know anything about the sample. I guess I could have asked. My guess, it was something like a high tunnel. That's my guess. So what's happening with a root knot nematode or any of your nematodes? So not just feeding on the roots, but these knots or these let's see if I can do that. These knots or these they kind of serve like galls. These cysts, they're encircling roots and they're girdling roots. So if a root is girdled, you don't have uptake up until to the upper plant parts. So it's like having no root at all. And so the female nematode was able to feed on the root, but now she has rendered this root dysfunctional. And since this root can't, or this plant can't utilize the root, the plant is starving. So this is what was happening. So anytime you see any kind of root damage, remember we talked about fungal root rots, we talked about oomycete root rots. This is the same thing. So this is root loss, and with root loss, you don't have the water and nutrient uptake to upper plant parts. So basically, your plant would be starving. You could see wilt. You could see just stunting and dieback and slow death if populations were low. Or if you had a lot of environmental stress like heat or drought, and that plant needed immediate uptake. So if transpiration is really high and it tries to do a really quick uptake and doesn't get it, you can see kind of a sudden wilt. And that's what would happen if, the weather kind of got really dry or really hot, say in August. July was hotter than August at this point. But that's when you'll kind of see that sudden wilt. So it depends on the actual population. Let me try to get you a few more views of this crazy root knot situation here. And I hope you never have a situation this bad. This is like as bad as it gets. I can't believe the plant made it this long before it actually succumbed to drought and starvation. Okay, so that was harder to see than I expected, so my apologies. I do have another root knot sample. This is a carrot, so this might be a little bit better for you to see. I'm going to come down. We're going to keep going on the Dino Light. And so we have some disfigured carrots, so if you can see. Disfigured carrots here. There we go. Let's see if I can get this one here. There we go. So just kind of disfigured carrots. These carrots were not growing. They were just kind of stunted and struggling. And a carrot, of course, is a root. So I'm going to come down just a little bit with my light, and I want to show you. We've got some knots. I guess I had it pretty good there. We've got some knots on the actual carrots. So you'll see, no, you can't see that one. Let's see, can you see this one? You can see knots, and you can see a knot here. All right, let me see what I can do. Okay, so you see the disfiguring, you see the stunting, and this is a knot right here. And it's a little bit easy. It's much easier to see in 3D. Then, of course, the carrots have all of these fine feeder roots. Here we go. They tangled up, and now it's easy to see. So you'll see the knots on the end of these little fine roots. How's that? All right, let me try to zoom in, try to focus it there for you. So here they are. Here are the knots on these fine roots, and they are, and here's one on the very tip of the root. So those are root knot nematodes. You don't know what you're looking for, you're never going to see it, right? So it's not easy as pie. Oh, there we go. You see the knots in there. So these are encysted females that has basically, she's basically turned into an egg sac. So encysted females can live in the soil or not live, she's dead, but they can remain in the soil until conditions become soft. And so when it rains and the soil gets softer, so she can remain there for years. Very important to know if you have populations and to manage by crop rotations, by rotating into non-host plants.
A request to cut through a knot, and what would that look like?
Ah, okay. Let me see if I can do that. Okay, so the request was to cut through a knot. Maybe we can do one of the big tomato knots. Let's see what we can pull off. Cut through a few knots. And I was not prepared to do this, so let me try to find some kind of clean ones, because if not, it'll just be dirty. Okay. See this big one? We'll see what we can cut through here. Where are my tweezers? Okay, I'm completely unprepared for this, so we'll just get it done. Let me see if I can hold it with my scissors. So you see the soil, you see what part was the soil. All right, I'm gonna zoom down. I know you can't see. So the white is the egg. See, these are the eggs. This is the encysted plant part. So kind of the plant and the female all kind of blend in together, but the white you're seeing is the egg. Let me see if I can cut another one for you. Maybe we'll get a good angle. Let me see here. So these are, so yeah, you can't see it here. So these are underdeveloped eggs. Let's see, let's try the carrot. Let's see what we can get out of this carrot. Okay, so these eggs decompose because we've actually had these carrots in the cooler for a while. Let's see if we can see the actual. So you can see the granulation. That was the egg. Those were eggs, so decomposed eggs. So they did not hatch. So we saw three things. We saw this is kind of decomposed eggs. They did not hatch. Okay, they did not finish developing, but they are eggs. This knot here is very immature. So this is the encysted female, but you don't see, you see what's going to be some eggs, and they're not really developed. Let's see what else. Oh, I made a mess here. And then this one, we see some immature but starting to develop eggs. I think, let's see, what is that? Looks like we've got some, can I go any further? No, sorry. If you see there's some kind of mites walking around there. So this is, so something is consuming this sample. Okay.
Would an option for heated high tunnel is affected by root knot nematode be growing flowers on tables or in baskets?
So there's a question about an option if you've got nematodes is to grow on tables. Absolutely. Absolutely you can, but technically that's not a high tunnel. And if it's not a high tunnel, then you have to consider what your requirements are. So if you had a cost share high tunnel, like with a soil conservation service or something, you cannot do that. But typically, if you have developed a very high population of nematodes, you probably had that house there for a while. So this is our best, this is probably our best out of the three, and I can cut some more if you want me to. So you see kind of this soft body, that is the female, so she is encysting, and these are the immature eggs. We saw the little mites or whatever kind of insect or arthropod walking around. It looks like they're feasting on this. So this is probably not going to be a viable knot. I can go through it. I can go through a couple more. Maybe at the end we'll go through a couple of more and see if we can find any different. See, these are mushy. Oh, yuck. Okay, these are mushy for some reason, so I don't know. Ooh, jackpot. So can you see the eggs there? Let me get it. I keep touching the photo button. I know you all see the eggs there. I expected this one to not give us a good view. You can see the eggs inside. There we go. And we still see those little mites crawling around in there, but they're probably consuming these eggs, so they're obviously not viable. But all of those little white dots, those are nematode eggs. This is root knot nematode. This particular sample is tomato, but root knot nematodes have a host of a range of hosts, including soybeans and a lot of weeds. And one thing about that, so any of you who work with nursery growers, if you're going to move bald and burlap stock into northern, into any northern areas, then you're going to have a phytosanitation certificate because a lot of weeds are hosts. Okay. All right, we've got 10 minutes left, and I didn't think I would take this long. I've got a sample of a Japanese anemone, and I want to show you foliar nematodes. When I opened up, I told you that nematodes were actually soil-borne pathogens. This is one of the few. There's a pine wilt nematode that affects pine needles, and this one is a foliar nematode. And I want to show you some things. This is just cool. So you're probably not going to run into a whole lot of this, but I'll show you some very classic symptoms. And then we will get them out. We're going to do just kind of like a little nerdy experiment. So foliar nematodes are sucking out the contents of plant cells. And as they consume what they need from one, which is the very dark, then they move to another. So you see some of the, where am I? Okay, so you see some of the really dark where the contents are empty, and then you kind of see a gradient in the next one. And that kind of varies. Oh, let's see. No, I'm trying to. So you see some of these, see all the different colors, all the different ranges. So there are varying degrees of damage by foliar nematodes. What I'm going to do is I'm going to use the dyno light and a backlight, and we're going to see if we can get nematodes out of these samples. So I'm going to cut across. I'm probably going to use this one. I'm going to cut across. I'm going to get myself some of the bad tissue, and I'm going to get myself some of the not so brown tissue, and I'm going to make a slide. And then we are going to look at, see if we can see some nematodes wiggling out. And that's just fun. We're just doing this for fun. Kim, can you do me a favor and hand me that glass of water over there? Thank you. So if you want me to switch out and you can see what I'm doing. So I just cut a piece of leaf tissue. So it's big for the size of what we usually use to make a slide. And I had a slide here. I was being prepared. And so this is my backlight. Remember my backlight that I had. Remember that I told you it was the $500 one. So I'm gonna put some water from my personal water glass on the slide. Set the piece of leaf on there, and then use a cover slip to trap it in. Now I'm coming under the microscope, switching out to share with you again. And I'm going to lower my stage. Sit out a little bit more. Right on the, oh, hang on. Sorry, guys. Trying to get my stage to lower. All right, I'm having problems here. Bear with me. Okay, tried it out yesterday so that I could make sure and get it clear for you. All right, so I'm going to another plane with my dino light. I'm just having problems moving this guy around. And this is when the nematodes wiggle out of the plant tissue. And they're not wiggling today. This is a great sample. Sorry. This was a really cool, this is like the coolest demo ever. And the easiest one, and I can't seem to get it for you today. The nematodes wiggle out of the plant tissue and they wiggle into the water. Okay, I know we're out of time. I'll try to get that for you and then put it on a video on the web because it's pretty cool. And so if you suspect foliar nematodes, it's something, this is what we would do in the lab. Okay, I'll redo this, shoot a video, see my water is evaporating. I knocked my cover slip off with the dino light. Okay, all right, apologies, we don't have wiggling nematodes today, but I will get them in a second for you. All right, so our time is almost up. Something I wanted to cover today was resources. We kind of went through some a little bit more complex of the pathogen groups. And so the question is, where do you start? So I wanted to cover this more extensively. Maybe we can do it in the practical series. So where do you start? If you've got a problem on a particular crop and you know what that actual host is, my first recommendation is either to go with a scouting guide of some type. We have lots of scouting guides from veggies, field crops. This one is high tunnels, fruit crops. So we've been working on these through our IPM series. They're always available, and I will just flip through one of them. So basically, it's a picture book, and you can get a little bit closer. If you just really don't know where to start, an option is to just do a Google search. Root diseases of carrots, root diseases of tomato, and then hit like Google Images and just see if you can find anything that might be kind of close. And sometimes it gets you a little bit closer, and from there, then you can jump into the literature, either that we provide, some of the fact sheets, or give you an idea of what kind of sample to submit. So guilty, I Google image search a lot, especially with ornamentals, because ornamentals are just hard. Every day is a new day. Every day we have something else. So using your resources, but knowing which resource to use is kind of that place where we get stuck. But the Scouting Guide series are really good because those are Kentucky specific, and we put the most common things there. And after that, you can go to more specific literature. But don't hesitate to Google search. Just know you can't stop with Google search. It's kind of like Wikipedia, right? It's your first search, and from there you move on to something that might be more specific. And that's what we will always be here to help you do if you have no idea. Any questions? Okay, so we are going to talk about the quiz. Kim is going to paste it for you. I'm going to share. Let's see, she made a handy dandy slide for you. This is the, it's not, oh, there, well, what is it looking like on your end? Are you seeing the double screen? Okay, all right, so, can you hit my escape button? Get out of this double screen. All right, sorry guys, we're trying sometimes to do these shares and it doesn't quite work out. So here is the quiz. Here is the link for the quiz. Kim is pasting it in right now for you to just copy paste it. Always, the training website is plantdiseasetraining.ca.uky.edu. You can click on webinars and all the webinars are there. All the videos are there. The surveys or the quizzes are not active for any of the old ones anymore. This one will be. It will be there for 1 week. The field practicum information is going to come later. So usually give us about an hour to an hour and a half after this webinar, and that's when the video will appear. But in the meantime, Kim has pasted, oh, she's having to reboot, yeah. Sorry. So this is where the survey can be found. Okay, Kim is going to do it for us right now. You're there. Okay, guys. Just give us a second. Look, the 6 links. Right about where you were. Up. There. All right. We're pasting it in. Oh, yeah. There we go. Jet, do the open. Yeah, there we go. This one. That one. No. Uh-oh. Oh, I know. Go back to PowerPoint. You have to stop share. Okay, now put it. Yeah, we're copying it, Amy. We're having difficulties. Okay, can I get my mouse back? All right. So the link is there, pasted in the chat box. So just copy and paste it where you are. And I am going to put it back up for the recording because the people watching this from a recording have to actually type it in because they can't see the chat on a recording. As usual, there are 3 questions, 3 content questions. One question that lets you leave comments. And the 5th question, you are required to put your name in so that you can get course credits. I think we are caught up on all of the other sessions, correct, Kim? And those have been sent to Kevin Horn and in Ricky Jurgen's office. So any questions? All right. Any other comments? All right, everybody. I know we went a little bit over time today. That was not my intention. My apologies. We will see you in 2 weeks, which is our final, our 7th webinar. And this one, for those of you watching the recording, you have 1 week to take the quiz and to get credit. So if you need anything from me in the meantime, let me know. I will be posting the link and the announcement for the part 2 of fungi. It's not required. It is optional, or it can be a makeup for the first fungi session if you missed it. So let me know if you have any questions there. I will be posting it, and it will be, where's my note? It will be held on the 31st of August is when we're going to do the kind of a makeup fungal session. So, and remember to register for your practical. We're closing that 2 weeks before each session. We're going to close registration for the practicum series. There are 4 across the state. All right. Yes, I am, Philip, I am going to record the final or the second fungal session. It will be just like this one, but if you got credit for the first fungal section, you cannot get credit for the second one, but you can watch it. You're absolutely welcome to watch it. You just can't get credit for it. There will be just whatever I find outside, so it'll probably be very different fungi, much of the same concepts, but just different examples. And there's so many fungi. We could do 7 sessions on fungi alone, and we could get something different every time. So I know several people were interested, so it will be open for everyone and for all agents, not just those in the certificate program. Okay, everyone, it was a pleasure. Happy Friday. 1 more to go. We can do it. We're on the home stretch now, and we'll see you in 2 weeks. Thank you.